So 12.2 is going to deal with the replication of the DNA, which we learned about during mitosis and meiosis, that the DNA is going to replicate during interphase. So prokaryotic cells lack nuclei, and many of the organelles found in eukaryotes. So their DNA is located in the cytoplasm, and most of them have a single circular DNA molecule that contains nearly all of the genetic information. This large DNA is referred to as the cell's chromosome. Right? Now, eukaryotic cells, in difference, are going to have 1,000 times the amount of DNA, and it's found in the nucleus. And diploid human cells have 46 chromosomes. So we, as eukaryotes, have more DNA, and prokaryotes don't have a nucleus. They don't have membrane-bound organelles. So because of that lack of membrane-bound organelles, they do not have a nucleus. So prokaryotes would be things like bacteria. Eukaryotes would be things like plants, animals, fungus. DNA length. DNA is surprisingly long, however. In a prokaryotic E. coli, which lives in the human colon, there are 4,639,221 base pairs, which is about 1.6 millimeters in length, which is pretty crazy how long that is. The DNA is folded over and over because the DNA is about 1,000 times longer than the bacteria itself, and the equivalent is 2,000 feet of rope jammed into a backpack. So if you wind it up nice and neatly, you could fit 2,000 feet of thin rope or string into a backpack. In humans, DNA is even more tightly packed. Human contains about 1,000 times many base pairs of DNA as bacteria. It means that the human nucleus contains more than a meter of DNA, so 1 meter, which is about 39 inches of DNA. So if we take a look right here, this is just showing the chromosome of bacteria. See how it's a circular loop? It's kind of folded over, so it's just a loop right here. And you see that there are still the base pairing rules. So even though they are bacteria, they still have the base pairing. Okay. The way it's so tightly packed is because of the chromosome composition. Chromosomes in eukaryotes contain both DNA and proteins packed together to form a substance called chromatin. All right. Chromatin is DNA that is tightly packed around proteins called histones, and the DNA and the histone create a structure called a nucleosome. Nucleosomes pack with one another to form a thick fiber, which is shortened by a system of loops and coils. The next one, that NU, should be nucleosome, which will be on the next slide. If we take a look at that right now. Nucleosomes are important because they're able to fold a large amount of DNA into a small space. Now, DNA replication here. Watson and Crick's discovery of the double helix shape of DNA allowed several of the questions about DNA to be answered. Each strand has all the information necessary for base pairing to occur. If you separate the two strands, base pairing would allow you to reconstruct the other. So replication is the copying process in which DNA is duplicated. During replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new strand. Right? So basically you just unzip it and then you add new bases in as you go along. In prokaryotes, DNA replication begins at a single point in the chromosome and proceeds often in two directions until the entire chromosome is replicated. In eukaryotes, however, DNA replication occurs at hundreds of places and proceeds in both directions until the chromosome is completely copied. So prokaryotes, since it is a circular loop, it goes in one direction until it loops around. But in eukaryotes, we have many, many different spots where it happens and it proceeds along. Now, replication forks are the sites where replication and separation occurs. Replication occurs by a number of enzymes unzipping or separating each side. DNA polymerase is used to get each individual nucleotide to produce DNA. It will also proofread each strand to ensure the perfect copy. So you have many different enzymes that will work together to separate the DNA. So you have helicase, which will unzip the DNA, ligase, which connects the two new bases together, polymerase, which adds the nucleotide, and proofreads. All right. So if we take a look right here, here would be the replication forks where the DNA is separated, and we add new bases in as we move along.